This protocol can be used to characterize non-muscle myosin dynamics at the protein level, which can inform the motile properties that contribute to their roles in cells. This is a fast, reproducible, and highly controlled method that can be used to study the effects of various regulatory conditions on myosin motility, informing their cellular behaviors. These techniques can be used to investigate how disease-causing mutations in non-muscle myosins affect their behaviors at the single molecule and ensemble level. The general approach of this methodology can be applied to various other cytoskeletal systems, such as the characterization of purify kinesins and dyneins with microtubules.
This protocol's versatility can raise questions about which fluorophores and chemical conditions are most appropriate, but the method can be optimized relatively quickly. Understanding how to set up and coat the flow chambers makes for a strong foundation upon which one can adapt various conditions for these experiments.
To begin, prepare a 1% nitrocellulose solution in amyl acetate and place a circular filter paper with a 125 mm diameter on the bottom of a tissue culture plate. Load 8 square cover slips onto a rack and thoroughly wash them with approximately 2 to 5 milliliters of 200 proof ethanol, followed by 2 to 5 milliliters of distilled water. Then dry the cover slips completely using a filtered air line or nitrogen line. Slowly pipette 10 microliters of 1% nitrocellulose solution along one edge of one cover slip. Smear it across the rest of the cover slip using the side of a 200 microliter pipette tip in one smooth motion. Then place this cover slip on the tissue culture dish with the nitrogen.